Hi, I'm Kahil. Welcome to Full STEM Ahead Long Island. You may not know it, but Long Island is a world leader in biology. Scientists and students here are making discoveries to improve how we treat diseases, make new biofuels from plants, and more. I'm at Cold Spring Harbor Laboratory. It's home to 8 Nobel Prizes, and over 1,000 people work here. Let's go see what's happening.
Cold Spring Harbor Laboratory has been here for over 125 years now. We are a vibrant research community, a lot of research labs, also a Center for Education. We have the DNA Learning Center here, a lot of conferences that come here to learn new things in biology and talk about biology.
What type of research do you do here?
I work in 2 areas. One is DNA replication, and I also work in an area called RNA interference. In general, what RNA interference is that when you express a gene is made out of DNA.
Right.
You transcribe it into something called RNA, and from RNA we translate that into proteins that are usually the workhorses in the cell and in our body. If you take a small piece of RNA that matches that intermediate RNA between the gene and the protein, you actually destroy that RNA and then the protein cannot be made. That is used in our cells just naturally, but sometimes that gets out of control. So for example, 70% of neuroblastomas are as a result of that little small RNA not being there when it's supposed to be. And so that causes the cells to replicate in an orderly way, and so that results in a tumor. And now, using our technology of structural biology, we were able to discover which protein actually it is that destroys that RNA. So now there is a new therapy that uses this type of process to our advantage.
I'm here with Bree Bebel, a graduate student.
Well, I've always been really interested in how things work, especially things in the body. So I went into college knowing that I wanted to study biology, but I thought I wanted to be a doctor. And we're using this textbook, and there was all these references about Cold Spring Harbor Laboratory, and it just sounded like this amazing place where so much cool science had happened. When I came here, I knew right away that this was a place for me.
What does the future hold for you?
I'd really love to be a teacher, especially for undergraduate students, and really get to share with them how to work in a lab and have them help me with my research experiments.
Before I came here, I was in Germany where I did my PhD. So I was kind of following Joshua Torr's lab and the kind of work she does here. But I feel like it's an amazing place to be. One, of course, it's in terms of science. Also, the nature, it's amazing here in Long Island.
So what's the next step for you?
Back in India, there are more people who kind of need this kind of information because it's still developing. If I can go back, I can probably contribute much more in Indian society, Indian scientific society.
Are there special kinds of tools that you use to do this work?
We use something called cryo-electron microscopy. We have a really high-end machine here at Cold Spring Harbor Laboratory, and that really enables us to look at particles of molecules and really study them in atomic detail.
Is this the only tool to use to do this kind of research?
We've actually been using X-ray crystallography for a long time, and we are really very, very fortunate to be close to Brookhaven National Laboratory and the National Synchrotron light source, the second one now that I've been using. It's really an amazing resource where we can get high resolution pictures of the molecules that we study. That's really, in some cases, the method of choice for us.
Limor, thank you for your time today. Now let's head east on Long Island's Innovation Corridor to Brookhaven National Laboratory, where Aleda will give us a close-up view of the National Synchrotron Light Source 2.
We are at Brookhaven Lab at the National Synchrotron Light Source 2. This machine is so big that Yankee Stadium can fit in it. Let's go inside and meet the people who use it.
So this machine uses a method which we call X-ray crystallography. This uses X-rays to determine this three-dimensional structure. And we get images not like a light microscope, but we get the images by having the X-rays scatter off the crystal of these molecules. These pictures are recorded on these kinds of detectors. They are like a big camera, and then they are transferred to a computer where we do the reconstruction of the electrons in these molecules which originally scattered the X-rays. That is the method by which you then obtain the three-dimensional structure.
So why is that important?
In a way, you can understand how these proteins function a little bit like playing with Lego. So they work by molecules fitting on top of each other or inside of each other or not fitting. A good example are antibodies which do or do not fit onto the outside hull of a virus to then help your immune system to attack them. Another good example are drugs. So when you want to inhibit a molecule and thereby kill a bacterium, you can do this by having a drug that blocks a very important function of this protein. Then this virus cannot replicate, and that way it's basically dead, which makes you healthy. Let me show you at the beamline. We have 2 students from Long Island high schools who are currently looking at the images that they took in their last shift.
So can you tell me a little bit about yourself?
Well, over the summer, I actually did a workshop at Brookhaven Lab. It was the structural biology workshop. I realized I have a love for structural biology, and I tried to find further programs that I could further the research in.
What kind of research you've been doing here?
We're researching anaplasmosis right now, and it's carried in ticks to cause diseases in humans. And by using X-ray diffraction, we can understand the structure and hopefully help cure these diseases. After grad school, I want to become a pharmacologist. Pharmacology is like a research into how drugs work and how they affect the body. Brookhaven Lab actually is what fueled my love for pharmacology, because before I even knew anything about Brookhaven Lab, I didn't know anything about pharmacology.
I'm currently researching the protein bovine thyroglobulin. It actually is very similar to this oligosaccharide, a compound called alpha-gal, and when transmitted by ticks, this compound can cause the red meat allergy in humans, which is very detrimental. At this time, bovine thyroglobulin has no known structure. We're hoping to find a structure so we can find out more information about how it interacts with other proteins and its function.
What have you learned by doing this?
First of all, we've learned basic lab skills like micropipetting, and to be more specific about protein crystallography, we've learned how to create crystals and fish crystals and even run the beamline. Before I started this research program, I was dead set on becoming a doctor, but now I'm gravitating more towards the research. It's such an honor. I never thought I would be in this place that I am right now. I love working with the scientists, and I've learned so much from them.
There are so many career opportunities in the life sciences here on Long Island, supported by world-class institutions like Cold Spring Harbor Laboratory and Brookhaven National Lab. To learn more, visit listemhub.org.